Ujú. Cuando vaya a empezar, pueden apagar la luz para que se vean bien. Intento de avanzar y repasar para que... No. A ver, ¿le hago? Sí. Igual tú me vas a decir del tiempo. ¿O cómo es? O si...
Plática.
Ujú.
Las pláticas se hace una ronda de preguntas. Eh, Después de eso son como alrededor de 4 o 5 preguntas, dependiendo del tiempo. Y se terminaría ya la dinámica.
Ok. Pero entonces tú me dices así como a los 40 o algo por el estilo, ¿o cómo?
Entonces, da tiempo. Y ya las preguntas son las que vamos modulándolas
Ok.
dependiendo del tiempo,
Bueno,
de cómo vamos con el tiempo.
ok, ok, está bien.
We're going to start. For the respect of the doctor, we're going to be in silence. We're going to talk about the relation of soil producer and human development with the honor of Dr. Cervantes. Dr. Cervantes obtained a degree in pharmaceutical biological chemistry from the Universidad Michoacana de San Nicolás Hidalgo. He began his studies in Senestat in the field of plant biotechnology. From 2006 to 2008, he conducted postdoctoral research at the Stem Research Center. From 2008 to 2011, he was a postdoctoral fellow at the Pacific Agri-Food Research Center in British Columbia, Canada. In 2011, he has been part of the Faculty of Natural Sciences at the Autonomous University of Querétaro. He has supervised around 2 doctoral theses, 10 master theses, and 15 undergraduate theses. His research focuses on the use of microorganisms for the development of beneficial products of agroindustrial development, understanding the molecular interaction in the infection process between corn rot or huitlacoche and mice, and the search for new antibiotics to combat frost-resistant bacteria. He belongs to the National System of Researchers at Level 2. Please give a big round of applause to Dr. José Antonio Cervantes.
Thank you. Okay, las veces, please. Hello to everyone. I'm very happy to be here with you, sharing what we are doing in the lab for several years. And I would like to thank, first of all, to the organizing committee for giving me the opportunity to be here with you this afternoon. So I'm pretty sure you are very related with this topic, the Agenda 2030. And as you know, we have a lot of things that we have to work with because the humanity is facing a lot of troubles. The troubles that I'm related with this agenda is with the number 2 goal, mainly I would like to know if you have any idea of how many people will be hungry within 10 years. Do you have any idea? How many people won't have anything to eat? Tell me a number.
2 billion.
2 billion of people all around the world? All around the world. Well, a little bit less. 660 million will face hunger in 5 years. So when this agenda started, they were planning that we will be able to solve this situation. But now it's very close. We just have 5 years left, and we still don't have idea, or we still have no the way to solve this situation. Another important fact is that 1 in 3 people will be in severe hunger, so even more than hunger. And this is a situation that is very important to take care about. And I got this information from this book, and I highlight some important things. The plants are suffering for biotic and abiotic stresses. As we can see here, 50% of the reduction of the agrochemicals are needed to do to alleviate the problems that we are making to the environment. So it means we are using a lot of chemicals to try to produce a lot of food. And that one is one of the problems that we do have. The other one is the antibiotic resistance. Is that someone can tell me what is the antibiotic resistance? Yes. Okay, well, the body is not developing the resistance, but the bacteria. So it means that we won't have any antibiotic to fix the problem. And that is a situation that is gonna kill more than 10 million of people in 2050. 10 million of people, more than any other disease. Cancer, 8 million. So what are we gonna do to try to avoid this problem? There are several bacteria, all of these: Escherichia, Staphylococcus, Klebsiella, Acinetobacter, Pseudomonas, and Enterococcus. All these bacteria like to live in hospitals, and all of them are resistant to antibiotic, or it's very easy for them to get resistance. As you can see, we have 2 big issues to try to solve. So what can we do to try to fix this problem that we created? As a research group, we start working with soil. We think that we have the ability to find something in soil that will help to fix the problem. What do you think that we have in soil that we can use to fix this situation? Someone tell me. What can we get from soil? Fungi. In general, we have a lot of microorganisms, right? So the soil is this area where we grow up a lot of food, obviously, and we do have several labels in this soil. We do have divided by horizons: horizon 0, horizon A, horizon B. The most important for us is 0 and A, and is the place where the plants grow up. But the plants are not alone. The plants are in relation with a lot of microorganisms. As you can see here, in 1 gram of soil, we can find all this amount of microorganisms. What happened? A lot of microorganisms. Thank you. We can find more than 100,000 fungi, and about bacteria even more, in just 1 gram of soil. We can find a lot of variety of microorganisms on soil. So we are thinking that we can find something useful in the soil to try to fix the problem. And as we can see here in this slide, we can talk about the microorganisms that are living with the plants. Yeah, we have a lot of microorganisms. As you can see here, we have a huge diversity. And later on, this tomato plant is selecting some microorganisms to start living with them. Later on, what happened when another microorganisms are introduced in the same area, because we used to add some organic matter that is full of microorganisms. And as you can see here now, the plant is able to share this small space with several microorganisms. Could be bacteria, could be fungi, etc. And we can modulate the population that we can find around the area of the plants that its name is rhizosphere. Okay, and in order to try to research about this situation, we select tomato to start making some developments to improve the production of tomato. In Mexico, this is very important crop because we have a gigantic diversity of tomato. As you can see, we have several shapes, and of course, each shape has different characteristics. Talking about the ability to avoid diseases, the amount that we can collect every season, and the quality of the fruits. In Mexico, due to the environment that we have, we can use the spring-summer cycle and the fall-winter cycle. So it means we can produce tomatoes during the whole year. And the question is if we are self-sufficient to produce a lot of tomato to feed the whole population in Mexico. And in this map, you can see that in almost all the states of Mexico, we can grow up tomato, almost all of them, and we are in the 10th place around the world. We use more than 50,000 hectares to grow up tomato, and we harvest more than 1 million of tons in the spring-summer cycle. So it looks that we produce a lot of tomato, and we are able to export tomato to other parts, mainly to US. In this chart, we can see that we have, or we make more than $2 million recently because of the tomato that we sent to US. And it looks like we are producing a lot of tomato, and as you can see here, we produce more than 200 tons during the month of March, but later on it reduced a little bit to September. So what happened with this production? How can we improve it? To improve the production, we have a big problem. This crop is susceptible to be or to get disease by more than 200 pests, and one of them is Fusarium oxysporum. Fusarium is a fungus. We can find this fungus almost everywhere. It's a saprophyte. He likes to stay close to the roots of the plants. It's host specific, and this is an important thing. It can survive for more than 30 years on the soil. So it means that it's gonna be resting on the soil, and later on, in 10 years, you start trying to put tomato there, and if Fusarium is there, it's gonna start again the infection of the plant. Let me show you the life cycle of this pathogen. As you can see here, we do have this cell structure that its name is microconidia, microconidia, and chlamydospores. They are living on the soil, very happy, as I said, for even 30 years. And what happens when they detect a tomato plant? They start the germination process. Later on, the mycelium start growing all around the cells of the roots. And later on, this structure of the plants that its name is the xylem. Through this xylem is the way the plants is getting the water to support the whole plant. Since this mycelium is growing a lot in this part, it's gonna make a clock. After this clock is made, so the water is not passing through anymore, and the cells of this part start dying, and later on we can see the first symptoms in the plant. As you can see here, the plant is getting yellow in color, so it means they are not making a lot of chlorophyll, so they cannot make photosynthesis. Later on, the plant is gonna die, and the disease of this pathogen is named wild disease. So, as I mentioned before, in that book, they tell us that the biotic and abiotic stress that the plants are suffering because of different microorganisms like fungi, bacteria, nematodes, insects, and virus, they are going to make a lot of bad situations in the plants, like increasing reactive oxygen species. They will make changes in the hormone synthesis, and the plants are going to be ready to get diseases. So, in order to avoid this situation, for a long time, we have been using a lot of chemicals. The most principal chemical is methyl bromide that we spread all around the field to try to kill the fungus. And in the year of 2014, it was prohibited. So we cannot use it anymore. But the thing is that when we stop using that chemical, a lot of money is lost. Taking as an example, in the US, yeah, they are losing a lot of money in Florida. So we have to find another choice. When we don't have another choice, so we have to work on it. What happened here in Mexico? Here in Mexico, we are still using a lot of chemicals. And the thing is that these chemicals are really, really toxic. But the regulation here in Mexico is different. So in 2007, we spread more than 50,000 tons in the whole country about this chemical. Now the situation is a little bit better because in 2020, we are using the half. But what happened in the neighbor state, Guanajuato, as maybe you know, Guanajuato is an state that they do have a lot of production of agriculture, and they are using a lot of insecticides, herbicides, and fungicides. So another situation important is here in Sinaloa, where they found a lot of people is suffering from neurological problems because of the amount of these chemicals that they are using. So something must be made because these chemicals later, sooner or later, they are related with the development of some kind of cancer. All kind of cancers that we develop could be related with these chemicals. So we have to stop spreading this because the agricultural workers are suffering these effects. In order to try to make something different, we think that we can consider to work with these bacteria, plant growth promoting rhizobacteria. We call them PGPR, and we can find them on the soil. There are several of these bacteria like Pseudomonas, Bacillus, Pimillus, Bacillus thuringiensis, Serratia, etc., that they are going to help us in some way or another. When I said that the plant is making a communication with the microorganisms, I was talking about this. As you can see here, there's a plant that is growing on the soil, and in this part, there's a lot of microorganisms, showing that we do have a huge biodiversity, and the root system is interacting directly with these bacteria. They are going to make a molecular communication that is very difficult to explain in a few minutes. But the thing is that these bacteria are going to make the plant express some defense mechanisms in order to avoid the attack of pathogens. And they are also going to help the plant to produce several hormones. As we can see here, the bacteria are producing all these activities that is going to help the plant, like the production of lytic enzymes, cytophores, solubilization of phosphate, the production of antibiotics, and they are going to fight with other microorganisms to protect the plant and the plant is gonna be safe. The plant is gonna be very happy because of the pathogens are decreasing and the good bacteria are increasing the population. And as we can see here, once the plant establish a good relationship with microorganisms, they are gonna help the plant to promote the growth, also to establish a mechanism of defense, modulate the production of pheromones, the nutrients availability, and also the stress tolerance. Because remember, plants cannot move. Plants have to face whatever situation they have, and they can make it easier if they are getting some help from microorganisms. And with all this information, in 2015, we started working on this. We went to Irapuato. We were there to try to get some soil samples from an orchard of strawberries. In this orchard, they don't use any chemicals. So we said, if they don't use chemicals, we are gonna find a lot of microorganisms. So it's gonna be nice if we can go there, get some plants, as you can see here. These plants will recover all the soil from here and start looking for bacteria in some culture media. As you can see here, we do have a lot of colonies here. We do have some that are different, and we select some of them and prepare our strain collection. This strain collection is around 200 bacteria that we keep it at -80, and we are working with them little by little. We start this, and in 2020, Alejandra Torres, an undergraduate student, was working in the lab, and she found some interesting bacteria. She found some Pseudomonas, Panthoea, and Varioborax. She asked the question to these bacteria if they are able to inhibit the growth of this pathogen. All of them are fungal pathogens, but we are working mainly with these guys, Fusarium oxysporum and Sclerotium rossi. As you can see here in the control plate, the fungus is able to cover the full plate, but what happens when we have the bacteria here? As you can see here, the size of the colony is less, so it means they are able to reduce the growth of these bacteria. With this in mind, we start doing some other experiments. Daniel Mendoza started working in the lab, and we analyze a collection of Fusarium that we have from Colima State. As you can see here, all of them some Fusarium. I just want to show you these two Fusarium oxysporum. And these bacteria that we have here, they are able to reduce the growth of this pathogen around 80%. As you can see here, the ability of these bacteria with this fungus is a little bit less. This is the experiment that we carry out. We prepared this agar that we named poison agar. And as you can see here, this is a PDA agar plate. We grow up the bacteria here over a surface of cellophane. After the bacteria grew up here, we remove that cellophane, and we have a petri dish that is containing a lot of secondary metabolites. Over this plate, we seed the fungus, and as you can see here on the control plate, the fungus is able to cover the plate, but under this situation is not possible because here we do have a lot of molecules that are able to kill this pathogen. With this information in mind, we said, okay, what is going to happen if we put together 2 or more bacteria? So we made these experiments and we prepared that we call consortia. On green color, we do have bacteria that make a positive interaction, so it means they can grow up together. And we prepared these 4 consortia in order to see if they are able to control the growth of this pathogen in a better way. And when we test this, we have a little bit improvement of the ability of this pathogen or of this bacteria to control the growth of this pathogen. If you remember, we have something around 60% here, and now we have more than 70%. So these consortia are working very good because they are controlling the growth of this pathogen. And the next question was, what are the molecules that these consortia are producing? So in order to answer that, Daniel started working with some organic solvents that he was using ethyl acetate and methanol chloroform and also acetate. He grew up these bacteria in the culture media, and later on he recovered the antibiotics that they produce. When he treated some conidia of Fusarium with this antimycotic, we found very good results because these molecules are able to avoid the growth of this pathogen. We use a different amount of this pathogen, as you can see here, and the green color is the control where we don't have any antibiotic. As you can see here, we have missing growth. So it means that these molecules that are present here, isolated with hexane, are able to kill the spores of this fungus. So we are having, or we are showing that we do have some antibiotic molecules. As I mentioned, we are interested in the interaction between tomato and the bacteria. And as you can see here, the next question was, are these bacteria are able to promote the growth of the plant? So to answer that question, first of all, what's important to know if in a petri dish, these bacteria are able to produce some molecules that will help the plant to grow up. We analyzed several molecules like cyanuric acid, production of siderophores, solubilization of phosphate, and in every green color, we have a positive reactions. So it means that yes, we can use these bacteria as a growth promoters. And the next question that Daniel answered was, what happened with tomato? And as you can see here, we have the development of some tomato plants, and this part is growing up better in the treatments when we compare with the control. When we are making a better situation for the plants, it's very good if they produce a big root system, because they are gonna be able to get a lot of fruits and to make them to arrive to the final stage. So we analyze this information by statistics, and effectively, they are important information because these treatments are improving the stems and the root system in the plants of tomato. The next question was, what is going to happen with the phenological cycle of tomato? As you can imagine, the time to when you put the seed on the soil and the seed start growing, later on you have the plant, the flowers, the fruits, it's gonna take some time, maybe around 60, 70 days. And what is gonna happen if we use this bacteria? As you can see here in the control, we do have a plant that is flowering. There's very normal, nothing to be admired here. But what happened when we, Daniel, use this bacteria? When we apply this bacteria, look, we do have the tomatoes. At the same time that the control plant is just making the flowers, in the treatment, we already have the fruits. So it's getting a little bit faster, the production of fruits. And we analyze with the other consortia, and we have similar results. We already have tomatoes when the control just have flowers. So that is very good because we can collect tomatoes faster, and we are gonna reduce a lot of money during every working day. The next question was, what happens if Fusarium is included in the plant? As I mentioned, Fusarium likes to attack this plant and he likes to kill tomatoes. So in this experiment, Daniel added some conidia, yes, on the plant. And the question was, what happens when conidia are present in the plant? As we were expecting in the control plant, the plant start dying. As you can see here, the leaves are not green anymore. The plant didn't grow enough, and later on, die. But what happened when we use these consortia? When we use these consortia, look, the plant looks very healthy. Of course, there are some leaves that are dry, but most of the plant is still green, and they are producing tomatoes. So we were very happy with this result, and also with the other consortia. So with this bacteria, we were able to protect the plant against the Fusarium, and we also were able to make faster the production of tomato. But we have a funny situation here. I forgot to tell you this. Also, the root system is really improved. As you can see here, the root system of the plants that we supply the bacteria, they are bigger, and they are able, as I said before, to keep stronger plants and taller plants and more fruits, of course. But the funny situation comes here. Now we are able to produce a lot of tomato, right? And what are we gonna do with that such amount of tomato? What is gonna happen if we are producing a lot of tomato at the same time? We have to have someone who is gonna buy them, or we have to keep them in some place. But what's gonna happen with, imagine this amount of tomatoes here, they are gonna get moldy very fast. So we were thinking, what is gonna happen if we use this bacteria to try to protect the tomatoes? And Daniel set up this experiment. He grew up this bacteria in a cultured liquid media. Later on, he removed the bacteria by centrifugation, and he prepared this cell-free extract. We do have here a lot of metabolites. We don't know what is in here, but when we put this tomato in here, later on, let stand that here for 3 hours. We here inoculate in these 3 spots the conidia of Fusarium, and we keep this tomato in this temperature, 28 degrees. What is gonna happen with these tomatoes? Okay, as you can see here, and as you can imagine in the control, where we apply the fungus, after 4 days, we have the full tomato cover with this pathogen, Rhizopus arizus, that is a post-harvest problem. We use as a control sodium benzoate, that is a chemical that we can eat, that we use as a food preservative, to see if sodium benzoate is able to inhibit the growth of this fungus, and is not able to do that. So the next question was, the cell-free extract is gonna help tomato for not to get sick? And fortunately, when Daniel got these results, we were very excited because he found that tomatoes are healthy. They stay in this situation for more than 30 days. So it means that here we do have molecules that we can spread on these tomatoes, and these tomatoes is gonna stay healthy for a long time. So we don't have to spend a lot of money to try to keep these tomatoes in a huge refrigerator. So it's gonna be a little bit cheaper even to move them from one place to another. Another very important fungus that we are working with is Sclerotium rolsi. He likes also to attack tomato and a lot of plants. Plants that are important for us because we eat them. And the thing with Sclerotium is this. You can see here this small small balloons that are brown color. Each one of these, the name is sclerotial. This is a petri dish where the fungus is growing. After 1 week or 2, we can see these small structures. Their name is sclerotial, and each of these is able to germinate and make more mycelium. As you can see here, this is a 50-cent coin, and this is a petri dish. When we collect this structure from this petri dish, we got here more than 1,000 sclerotia. So they produce a lot. And the problem with this is that they can stay for 30 years on the soil, ready when there's another plant to be attacked. So it's like if they are seeds. So in order to try to work on offer a solution, we start working with this bacteria, another set of bacteria. And as you can see here, in the control after say 5 days of growing, the control is growing up. But what happened with the treatment with this consortia? We can see here that we stop the growing of this fungus. And after 30 days, at the time we collect the sclerotia, as we can see here, it's hard to see this structure because this consortia was able to inhibit the production of this. So we have a very good antifungus. We also prepare a cell-free extract, put some extract to these sclerotia, put them on PDA plate to allow them to grow. And as you can see here, I hope you can see these small dots. These are the sclerotia that they were not able to germinate. So it means we kill them with these compounds. As I said, we still don't know what is in these compounds, but there's something very important that is able to kill these microorganisms. And as I mentioned at the very beginning, we are interested in the other situation, that is the bacterial resistance. So in 2020, we went to Wisconsin to work in this lab with Joe Hendelson, that she started this networking. Its name is Tiny Earth. She used to work during Obama's government, and she said that we have a real problem with this bacteria, that we have to do something to try to stop them. And the idea of this Tiny Earth is work with soil, work with undergrad students, and try to find some new molecules. Till now, they do have more than 26,000 bacteria that they found in different kind of soils all around the world. As you can see here, we are working here in Mexico, and there are people working in this network in all the world, all because of the same idea, try to find new antibiotics. Then we ask if these bacteria are able to produce some antibiotics that are able to kill another bacteria, but most important, to kill resistant bacteria. We are interested in these bacteria. With this in mind, we start looking for some bacterial soil, and we prepare our collection of microorganisms. We went to several parts of Querétaro and find some soil. And we test these bacteria with some bacteria that are pathogens, like Staphylococcus aureus, that is an important pathogen. And as you can see here, this part is telling us that this bacteria and this bacteria are able to inhibit the growth of this Staphylococcus. And on this test, as you can see, this inhibition helus also is telling us that the bacteria are able to inhibit the growth of another bacteria. With this information of mine, we prepare some organic extracts using hexane, using ethyl acetate. And as you can see here, we can get some incubation halus that are very big. This is the control, is the solvent. And as you can see here, the solvent is not killing the bacteria. All the green color, it means that these bacteria that they found, they are able to kill these bacteria. We do have good results because we are able to kill Staphylococcus, some other Enterobacter, and Bacillus. Also, this bacteria, Klebsiella pneumoniae, as you can see, only two bacteria, two extracts, are able to kill them. So we got all of these bacteria from soil. So the question is, who is this bacteria? So we carry out the molecular identification using the sequence of the 16S DNA. And in this case, this bacteria is serratic. So we are working now with these bacteria. We do have several to try to get the antibiotics that they produce. And later on, we are going to make some devices to put them in a bin to use them to kill this kind of bacteria. So with this, I just want to say thank you. And all these members on the lab have been working there for a long time. Here I have Alejandra. She started with this story like 7 years ago. Later on, some other members arrived to the lab. Daniel Mendoza was working a lot with these bacteria and tomato plants. Miriam Cordero, she got her master's degree 2 months ago, working with Sclerotium. He do have Adrian Salcedo. He's working with this cell-free extract to make nanoparticles to kill some microorganisms, particularly Fusarium oxysporum. Mariana, she's working also with some microorganisms, in this case, some fungus, to kill bacteria that are resistant to antibiotic. And this is Felix. He is working, trying to develop a plant biostimulate. So with this slide, I finish my presentation, and I hope you have some questions to be answered. Thank you very much.
Thank you very much, Doctor. We are going to open the floor for some questions. Someone have?
Is it me? Oh. Oh. Yo lo oía y lo oía.
Hello, Doctor. Thank you for the talk. That was a very interesting conference. I wanted to know by which modality you added the microbes to the soil for the tomatoes. Like, is it in a specific way, or just how?
Okay, when the plants are more or less about 1.5 months of age, like this size, we collect the spores from a petri dish. We count them and we prepare a solution. And later on, we put these bacteria, these spores on soil because this pathogen likes to stay on soil. He starts the infection through the root system. So it needs to be close to the root system in order to start growing up and start the penetration through the root.
Okay, so you put it close to where?
To the stem.
Okay.
Yeah.
Yeah. All right. Thank you.
Yeah, in liquid, just in water.
Okay. Thank you.
So some questions.
Hi. Thank you for your talk. I wanted to know, like, how do you plan to know what are the compounds that are in the extract that you mentioned?
Yeah, that is the next step. Definitely, we need to find out what molecules are present there in order to be able, because of the regulation system, to spread them. Well, the easiest way is to make HPLC to start discovering what molecules are present there. And later on, we have to find out if we can use that molecules, because remember, we are trying to apply the tomato. And what is going to happen if one of those molecules is not very good for the human. But we can always wash the tomatoes before we can eat them, and maybe we can wash away these molecules. But there's something that we have to work with very hard, and Daniel Mendoza is here, and he's in the PhD, and I hope he found this.
Okay, thank you.
Hello, Dr. First of all, thank you for the presentation. It was very interesting. And I'm curious about the cost of production for the tomato industry. Would it decrease or increase, or if it decreased, for how much? Would be it affordable for the small companies?
Yeah, that is something really important that we have to start working very hard on that. Because if we are trying to apply this, should be something cheaper that we do have now. But if we consider that we put just the tomatoes in this liquid and remove the tomatoes, we are not using a lot. Yeah, because we just put them 3 hours and remove them. The tricky thing here is how are we going to apply to a lot of tomatoes. We have to develop maybe some sprinkles or something that spreads the tomato in a big room. The tomatoes are passing through a van. They receive this solution, and later on they go to the next step, that is drying. But I think I'm pretty sure it's gonna be really affordable, because if you are a producer from Sinaloa, for example, that they produce a lot of tomato, and they have to send these tomatoes here to Mexico City, or maybe to send them to US. It means a lot of time traveling in a truck, and you have to pay a truck with refrigeration. How much is the cost of these trucks? I have no idea, but I'm pretty sure it's not cheap. So if we can send these tomatoes after we spread this solution in a regular bus, in a regular truck without the freezing system, I'm pretty sure it's gonna be easier. And besides, we are interested to apply this to another fruits, like, for example, berries, because berries are very expensive. And most of the production here in Mexico goes through the other countries. And they are very easy to get rotten in the way. So if we are able to apply this one there, I think it's gonna be a success.
First, thank you for your talk. I also thought that it was a very interesting topic. And I have two questions. First, like, what do you think? Well, maybe you answered them with both of the questions that they asked you. What was like the greatest challenge that you faced when performing like this experiment? And also, do you think this will work out in other type of crops, or depending, or specifically, or what will be like other?
Yeah, we already have some information with this. Daniel started working a long time ago with this, also with pepper, with pimiento pepper. Pimiento, we also send a lot of tons to US. So it's gonna be more or less the same situation, because they belong to the same family. And we can use this kind of technology, biotechnology, with another plants like cucumber, for example. The only thing that we have to do is start testing in several kind of plants, but for sure it's gonna work. And besides, we already know that works with Fusarium and with Sclerotium. And these two pathogens, they are able to infect around 200 different kind of plants. So they are a little bit related. So it means that we can kill or control the population of these pathogens. We are very confident that it's gonna work in some other systems. We just have to make another experiments to have the results.
Someone else has other question? Okay.
So, again, thank you for your talk.
Thank you.
It's really interesting to learn that there's labs that are doing all this. So I was wondering, do you have any guess as to the effects you describe on phenology? Like, why is that happening? Because most of the other stuff, I guess, makes sense in the context of antibiotic activity and so on from the bacteria, but why would it accelerate the cycle?
Yeah, our guessing is that all the genes that are related with the flowering process, for example,
Uh-huh.
are upregulated with these molecules producing by the bacteria. Now, Daniel is going to start working with RNA.
Mhm.
He's going to ask to the plant what happened when we add these bacteria to these genes. Are they upregulated or not?
Uh-huh.
But our guessing is that this is a regulation about the ability to modulate the expression of these genes in order to produce more amount of hormones, phytohormones.
So to some degree, it might be like manipulation of the plant by the bacteria.
Yeah, when the bacteria start the communication with the plant, they are talking each other and they are telling us, I can give you some molecules that you need. The bacteria said, yes, I will take them and I'm going to start growing up because I'm going to eat them. And I'm going to make that you change the way you use your genome, the way you use your genes, and you can start expressing a lot of genes at the same times. And the results, what is going to be? That the plants start growing faster and developing faster. And it seems that everyone is happy with this situation, but also something that we have to do is to test the quality of the tomatoes. We haven't that done yet, and we have to know if the tomatoes, we can see that they can stay for a long time. The shelf life is very good now, but we still don't know what is the characteristic of these tomatoes.
You mean like nutritionally?
Nutritionally, we have to ask, and also the flavor, because if the flavors change, or maybe the population, the people is not going to be very happy to buy them. But we have to do a lot of things before that. We have the permissions by Cofepris to let us sell them, but we are working on that.
All right. Excellent. Thank you. Anyone else?
Well, from part of the STEM community and our sponsors, we want to thank you with a small gift. Thanks a lot, Dr. Cervantes. Thanks for the knowledge, for your presentation. Yeah, let's give him another round of applause, please.
Thank you. Thanks. Thanks a lot.
There is a round table at 5 p.m. if someone you want to be here. And well,
Yeah.
we see you next.
It's technology for sustainability at 5 p.m. We have a little break right now. As always in these breaks, you can visit the Axolotarium for the next half hour. The doors are open.